Let's start. Hello, everyone, and welcome back after our summer break to PANIRD. So tonight we'll talk about CRB1 gene mutation. Our moderator is Dr. René Moya, and he had a great presentation last week. He gave us a little bit of headache, to think about, which gene is, there and what we need to do additionally. So today our presenters will be Dr. Moya together with Dr. Rojas and Dr. Perez, and our host is Dr. Mendoza. So I just would like to tell you a few words about each presenter. So Dr. Alfredo Rojas is ophthalmologist. He's ophthalmologist from Chile, and he finished his ophthalmology residency in University of Chile, and after that he did fellowship in Retina, and he's working in University of Chile since 2020 until now.
Okay. Do you see my screen?
Yes.
Okay. Well, hello everyone. My name is Alfredo Rojas, and I'm a Retina Fellow in Santiago, Chile, and I'm very, very glad to be here with you today. I'm going to talk to you today about a general view of the CRB1 gene, and I hope you can learn something else about it. The story begins with this fly called Drosophila melanogaster, that has been very important to understand more about this gene. This is because the CRB is a large transmembrane protein initially discovered at the apical membrane of the Drosophila epithelial cells. And many years later, it was found that mutations in a human homologue of the Drosophila, denoted as CRB1 or Crumb's homologue 1, was involved in retinal dystrophy in humans. And why we want to know more about it? Because it's estimated that near 80,000 patients worldwide are or will become blind in their first decades of life due to mutations in CRB1 gene. Also, these mutations are the second leading cause for leber congenital amaurosis, with 10,000 patients affected around the world and accounting for 10% of cases of this disease. The mutations in CRB1 also cause early and adult onset retinitis pigmentosa, accounting for 3% of cases of all RPs and 70,000 patients worldwide affected with this phenotype. There are more than 200 different mutations described along the CRB1 gene, resulting in different retinal phenotypes, predominantly with autosomic recessive inheritance, but without a clear genotype-phenotype correlation. The clinical variability supports the hypothesis that the phenotype of patients with CRB1 mutations is modulated by other factors that we are going to talk later. The main two phenotypes of the CRB1 mutation are the leber congenital amaurosis and retinitis pigmentosa, and this one with different subtypes like early onset RP or codes like or P between others. Here we can see the human CRB1 gene that is located in the long arm of the chromosome 1 and contains 12 exons, has 12 identified transcript variants so far, and over 210 kilobases of genomic DNA. This protein has 1,406 amino acids with 19 epidermal growth factor-like domains, 3 laminin A globular domains, and a short intracellular domain of 37 amino acids. And this CRB complex or CRB family consists in 5 CRB proteins. In mammalian tissues, CRB1 and CRB2 are predominantly expressed in the retina. However, the CRB2 expression is also found in other tissues, such as the kidney, the heart, the brain, and also the spinal cord. In addition, unlike CRB1, CRB2 also localize in the retinal pigment epithelium. Mutations in CRB1 cause visual impairment, as we know, and in the other hand, the CRB2 mutations are also associated with ventriculomegaly, hydrocephalus, and renal disease, ranging from a focal segmental glomulosclerosis to congenital nephrosis. The CRB3 has not yet been associated with human disease. Here we can see the structure of all the CRB family, and you can notice that the CRB2 protein displays almost the same protein structure as CRB1, except at the depletion of 4 EGF domains. CRB3 and 3b, they lack the entire typical extracellular domain, but contains the transmembrane domain next to the intracellular part with 37 amino acids that is similar to the CRB1 protein. More research is required to define the function and more characteristics of the recently described CRB1b isoform. And where is the localization in the retina of these proteins? The principal interaction of the CRB complex is with photoreceptors and Müller glial cells. In the picture of the left, we can see the photoreceptors in red and light blue and Müller glial cells in orange. All this happens at the level of the external limiting membrane in the subapical region in both cellular types above the adherent junction. The CRB complex could be between photoreceptors, between Müller glial cells, or also between photoreceptors and Müller glial cells. Now, in mouse studies, have shown that CRB1 protein is only present in Müller cells, while CRB2 is present in both photoreceptors and Müller cells. And the first data in postmortem human retina revealed a different localization, where CRB2 is only located in Müller cells and CRB1 is located in both. However, recently it was shown that CRB1 and CRB2 are located in both cells at the subapical region in the second trimester of human fetal retina, but in the first trimester only CRB2 was detected. In summary, according to all this evidence, the hypothesis is that in humans, the CRB1 and CRB2 are located in both cell types at the subapical region. Therefore, both cells should be targeted to prevent retinal degeneration in RP patients. If you want to learn about the function of these proteins, we have to know that research studies have shown that CRB1 and CRB2 are apical polarity factors, and apical basal cell polarity is essential for the formation and function of epithelial tissues. The principal functions of the CRB complex is the cell proliferation and the cell adhesion and morphogenesis. About the cell adhesion and morphogenesis, we have 3 scenarios. In the first one, the core CRB complex is formed by the interaction of CRB and protein associated with Lin7-1, Pulse1, also known as MPP5. The ablation of MPP5 in mouse retinal pigment epithelium causes early onset retinal degeneration, while ablation of MPP5 in the neural retina does not. This suggests an essential role of Pulse1 at the tight junction of RPE, but not in their neural retina. In the second scenario, the binding of Pulse1 and CRB can lead to the recruitment of PATJ, or multiple PDC domain protein 1, or MUPP1, to the apical membrane. The PATJ connects and stabilizes the apical and lateral component of tight junctions in human. Finally, in the adult retina, the protein EPV4.1L5 shows substantial overlap at the external limiting membrane with CRB1 and Pulse1. When there is an overexpression of this protein, that affects the tightness of the cell junctions and results in disorganization of the tight junction markers. About the cell proliferation, in Müller cells, the YES-associated protein 1, also called YAP or YAP, has an important role in the damaged retina because it's a core member of the Hippo pathway, which regulates several biological processes, including cell proliferation and survival. When the CRB expression levels are low, the Hippo pathway is inactivated, YAP is not phosphorylated and can move to the nucleus where the YAP target genes are expressed, leading to increased cell proliferation and decreased apoptosis. In addition, the CRB1 and CRB2 deletion also lead to YAP signaling dysregulation, and all this data suggests an essential role of the CRB and the Hippo pathway in the control of cell proliferation. Over the years, there are multiple models developed mimicking the CRB1 related phenotype in patients. There are two principal models of investigation: the human derived retinal models with induced pluripotent stem cells with retinal organoids, and the animal retinal degeneration models. The retinal organoids from pluripotent stem cells achieve defining the localization and onset of expression of the CRB complex. Several members of the CRB complex, like CRB2, PULS1, PATJ, and MUPP1, all of them were detected at the external limiting membrane very early in the development of the organoids. Also, the onset of CRB1 and CRB2 were observed in these models with a clear onset of CRB2 expression before CRB1 expression, like we talked before. In the animal models, the double retinal knockouts of CRB1 and CRB2 have helped to understand the contribution of the two CRB proteins to the retinal disease. 12 CRB1 RP-like mouse models have been described so far that are in this table, with different combination of ablations, different phenotypes, onset, and severity. In summary, the difference in phenotype onset between all these mouse models might be explained by the onset of the CRE expression during retinogenesis and by the cell type specific ablation. For example, ablation of CRB2 from a later time point result in a milder phenotype. Interestingly, when CRB2 is ablated in Müller cells, a very mild morphological phenotype with no functional consequences measured with ERG was observed, but ablation of both CRB1 and CRB2 from Müller cells caused a severe phenotype. All this data suggests that CRB2 has a redundant function in Müller cells, while in photoreceptors, it is essential for proper retinal function. About the treatment, no treatment is currently available for the treatment of CRB1 retinopathies. However, preclinical studies performing mice demonstrated the efficacy of adeno-associated virus as a treatment for CRB1-RP. These studies have shown that the expression of CRB1 was deleterious in CRB1 mouse models, but not in the wild-type mice. The strategies using functional family members from CRB2 to rescue CRB1-related retinopathies restored retinal function and structure in CRB1 mouse models. In that study, there was an improvement of the photoreceptors layer morphology and in the ERG response after the CRB2 delivery. Therefore, testing CRB1 and CRB2 expressing vectors in retinal organoids will help in the discovery of the CRB gene therapy treatment for CRB1-related retinal dystrophies in patients. In addition, there are different non-viral mediated approach for gene therapy. An example is the use of new technology using nanoparticles. In theory, these approaches have an unlimited gene packaging capacity, which display a big advantage for larger genes such as CRB1. These nanoparticles are also able to infect RPE and photoreceptors and can drive gene expression on a comparable scale and longevity than adenovirus in mice. The nanoparticles have a tolerable safety profile and is non-toxic in mouse and primate eyes. But more research is required to define the clinical relevance of these nanoparticles in retinal gene therapy. Finally, with current approaches, the optimal therapeutic window is missed in most patients. Typically, an ophthalmologist becomes involved only after the onset of retinal degeneration and vision loss. Under ideal conditions, a patient at risk for developing an inherited retinal degeneration will be identified well before disease onset and the start of vision loss. This will enable the clinician to intervene within the therapeutic window, providing a well-planned and personalized intervention. This is what we should fight for every day. Thanks.
Excelente.
Thank you very much, Alfredo.
You're welcome.
Thank you very much, Alfredo. Excellent presentation.